Here in lesson 2 on the skeletal system and bone tissue, we will conclude by looking at the process of bone formation, followed by bone remodeling or changes throughout life, as well as the factors required for bone growth and development, and as usual, conclude with the effects of aging on bone tissue. Let's move on to describe how bone tissue is formed. The process by which bone is formed is called ossification or osteogenesis. Throughout life, bone formation occurs in 4 situations. Initially, we need the development of the bony skeleton during embryonic development, so that's in uterus. Then, we need continued bone growth and development throughout infancy, childhood, and adolescence until adulthood when we stop growing. Thirdly, throughout life, remodeling of bone occurs where old bone is broken down and replaced by new bone. Particularly, we have breakdown of old bone in areas of decreased stress or to replace blood mineral levels from bone reserves, while bone buildup occurs in areas of increased bone stress where increased bone density is needed. And lastly, ossification is required for the repair of fractured bones. Ossification, again, that is the formation of bone tissue, actually begins during embryonic development at about 6 weeks gestation. Bone tissue develops from mesenchyme, which is one of the 3 embryonic tissues. So mesenchyme gives rise to all the connective tissues, including bone tissue, along with all the muscular tissues. Bone formation from mesenchyme follows one of 2 patterns, either intramembranous ossification or endochondral ossification. Intramembranous ossification is responsible for the development of a few bones in the body, including the flat bones of the skull, along with the mandible, part of the clavicle, and a few other bones. Endochondral ossification is the more common way bones are formed, particularly the long bones of the body. Let me give you a quick summary of each pattern of ossification. Intramembranous ossification occurs when mesenchymal cells are stimulated to differentiate into osteogenic cells, which then mature into osteoblasts. The osteoblasts secrete the organic extracellular matrix of osseous tissue and become surrounded by it, developing into the more mature osteocytes residing in lacunae. Following secretion of the osseous extracellular matrix, there is deposition of calcium and other minerals into the matrix, a process called calcification. This makes the matrix hard and rigid. Trabeculae then form within the bone tissue, and blood vessels develop in the spaces between the trabeculae. You will hopefully recall that trabeculae make up spongy bone. Finally, a thin layer of compact bone develops superficially around the core of spongy bone, and surrounding the compact bone, the remaining mesenchyme differentiates into the periosteum that surrounds the bone. So again, if you think of flat bone as a sandwich of spongy bone between 2 layers of compact bone, you can get an idea of how these bones come to be through intramembranous ossification. Now within the skull, the bones do not fit together perfectly, and there are spaces between the bones called fontanelles or soft spots. These spaces allow the flat bones to actually slide over each other to decrease the infant's head size to ease passage of the fetus through the birth canal during delivery. These spaces then become ossified after birth to complete formation of the skull. We will discuss endochondral ossification on the next slide. All right, on this slide, we just have a step-by-step overview of the process of endochondral ossification. You can see that after the mesenchymal layer is converted into a cartilage model, you can actually see the beginnings of the epiphyses and the diaphysis. The cartilage becomes calcified and begins to break down. Nutrient arteries enter the area, creating ossification centers, Most of that cartilage is then replaced with osseous tissue with the activity of the osteoblasts. Then more arteries infiltrate the ends of the model into the future epiphyses, forming secondary ossification centers. Please note that there's a band of cartilage that remains, and this cartilage forms the articular cartilage that covers the epiphyseal ends, as well as the epiphyseal plate that runs through the metaphysis. And this epiphyseal plate of cartilage is important for long bone growth, as we'll see in future slides. So the diaphysis is converted mainly into compact bone, while the epiphyses develop spongy bone. All right, so how do bones grow? Long bones grow in length or longitudinally by endochondral ossification. Bones grow in length due to first, growth of the cartilage in the epiphyseal plate and articular cartilage, which is then replaced by osseous tissue. Chondrocytes in the cartilage nearer to the epiphyses proliferate and extend the length of the bone towards the ends. Meanwhile, calcification of the older cartilage is followed by ossification as osteoblasts and blood vessels invade the deeper cartilage from the diaphysis. So, as the cartilage grows, the bone lengthens, and then the cartilage is replaced by bone tissue. So here's another summary of bone growth during infancy, childhood, and adolescence. So the cartilage that remains in long bones, forming the articular cartilage and the epiphyseal line, contain chondrocytes that proliferate and essentially extend the bone by extending the cartilage. But that cartilage becomes calcified, and then osteoblasts invade the area and lay down bony matrix, replacing the cartilage with bone tissue. So this is very similar to what we saw with endochondral ossification during fetal or embryonic and fetal development. Now, this continues throughout childhood and adolescence, and as an individual approaches adulthood, they stop growing in height because the cartilage of the epiphyseal plate is replaced by bone tissue. So it becomes ossified and is now called the epiphyseal line. You may hear the expression that the epiphyseal plate closes. So the chondrocytes are replaced by bony tissue. It was the proliferation of the chondrocytes that caused the bone to grow. And when the chondrocytes are no longer there and have been replaced by osteocytes, that now ends growth in height, and we reach our adult height. While bones are growing in length, they're also being remodeled to get the correct length and shape of both the diaphysis and the epiphysis. So let's talk about remodeling. Bones not only grow in length, but they also grow in width in a process called appositional growth. If your bones did not grow in width as they grew in length, you essentially would have human-sized toothpicks for your skeleton. During appositional growth, osteoblasts on the inner surface of the periosteum lay down new bone tissue. This increases the size of the bone from the outer surface. Concurrently, osteoclasts in the endosteum, which lines the medullary cavity, eat away or resorb old bone from the inner surface, widening the cavity. In this way, the bone increases in diameter while the thickness of the diaphysis wall is maintained and the bone does not become too heavy. As stated earlier, remodeling of bone occurs throughout life, from embryonic development and during infancy, childhood, and adolescence, throughout adulthood, and until death. There's an ongoing replacement of old bone tissue with new bone tissue, again, mainly in response to stressors on the bone, as well as blood mineral requirements. Osteoblasts build new bone in areas of increased stress, for example, where tendons attach to and pull on bone, especially in response to increased physical activity or even weight gain. Osteoblasts break down or resorb old bone in areas of decreased stress. For example, if one becomes sedentary or immobile. We will also consider the activity of these cells in response to blood calcium levels. Now, I mentioned in an earlier slide that the femur you have today is not the one you had a few months ago. Fun fact, the distal portion of each femur is replaced on average every 4 months. Normal bone metabolism, that is the buildup and breakdown of bone tissue, depends on several factors, which we will summarize here. Number one, minerals. Bone tissue is unique in that the extracellular matrix is highly mineralized. Large amounts of calcium and phosphorus, in addition to smaller amounts of magnesium, fluoride, and manganese, are required for bone growth and remodeling. Now, maybe some of you saw fluoride and thought, teeth! Yes, you need fluoride for strong teeth also, but please note that teeth are organs of the digestive system, not the skeletal system. All right, normal bone metabolism also requires adequate amounts of several vitamins. Vitamin A, in addition to being necessary for good eye health and vision, is also required to stimulate the activity of osteoblasts. So vitamin A is important for building new bone. Vitamin C is very important for bone health. Vitamin C is needed for the synthesis of collagen. You may have heard of a disease called scurvy. It was very common among sailors and pirates where they would become weak, and it was characterized by their teeth falling out. And they realized that the connective tissue in the gums that held the teeth in the jaws was breaking down because of decreased vitamin C and decreased collagen synthesis. So again, you need vitamin C for synthesis of collagen throughout the body in all of your connective tissues, but also in the bone. And recall that collagen is required for bone flexibility and also provides the framework for the deposition of the minerals we mentioned above. All right, moving on to vitamin D. Vitamin D helps build bone by increasing the absorption of calcium from the food we ingest in the gastrointestinal tract. And when we get to the integumentary system, we will see how vitamin D is processed in your body, but you definitely need vitamin D for calcium absorption. Otherwise, the calcium in the food you ingest will pass through the digestive tract unabsorbed. Finally, vitamins K and B have other purposes in the body, but in the skeletal system, they are needed for the synthesis of bone proteins. Proper bone homeostasis also requires hormones. Now, during infancy and childhood, the hormones that are most important to bone growth are called insulin-like growth factors, IGFs, and these are produced mainly by the liver in response to the hormone, human growth hormone, from the anterior pituitary. So human growth hormone exerts its effects mainly through insulin-like growth factor with respect to growth, particularly in the skeletal and muscular system, where here the IGF stimulate osteoblasts and promote cell division at the epiphyseal plate. So that would be the proliferation of the chondrocytes that allow for longitudinal growth. And insulin-like growth factors also enhance protein synthesis in bone tissue as well as muscle tissue. So, as the name suggests, these factors promote growth by stimulating the growth of bone and muscle. Thyroid hormones also are important during childhood development as they promote bone growth by stimulating osteoblasts as well. And insulin promotes bone growth by increasing the synthesis of bone proteins. Insulin is an anabolic hormone that promotes, among other things, protein synthesis. So remember, bone is not just minerals, it's not just calcium. Bone is 25% proteins. Other hormones begin to exert their effects at the beginning of puberty. So as one enters adolescence, the sex hormones, estrogens and androgens, testosterone being the main one, cause dramatic effects on bone growth. and are responsible for the sudden growth spurt that occurs during the teenage years. In females, estrogens promote changes in the skeleton, such as widening of the pelvis. In males, we see the effects of testosterone as it causes broadening of the shoulders and chiseling of those facial features. Now, interesting fact. The adrenal glands of both males and females produce androgens, and some tissues like adipose tissue can convert androgens into estrogens. This means that females produce both female and male sex hormones, estrogens more, and a little bit of androgens. Conversely, males produce a lot of testosterone, mainly from the testicles, but men also have circulating levels of estrogens. Hope that didn't blow your mind. Now, the estrogen in males and females is responsible for the closing of the epiphyseal plate. Because females produce more estrogens, mainly from their ovaries, the epiphyseal plates close earlier in females and they stop growing at an earlier age. Around 17, that's when females reach their adult height. While in males who produce less estrogens, their epiphyseal plates close later in life and males reach their adult height, stop growing at around age 19, 20, 21. Post-adolescence in adulthood, estrogens and androgens contribute to bone remodeling, mainly by slowing the resorption of old bone. So they work to maintain bone. This effect is more obvious in the older female, as the rate of bone breakdown outpaces that of bone buildup once estrogen levels fall significantly after menopause. The older female has a significantly higher risk of developing porous, low density, fragile bones in a condition called osteoporosis, and we will discuss that later. Other hormones, such as parathyroid hormone, calcitriol, which is the active form of vitamin D, and calcitonin, these hormones affect bone remodeling as well, but mainly their role is to regulate blood calcium levels. Let's look at that in the next few slides. One of the less obvious roles of bone tissue is that of calcium reservoir. The levels of calcium in the blood are actually maintained by balancing or altering the rates of calcium resorption from old bone and releasing that calcium into the blood versus the rates of calcium deposition from the blood into new bone. Now, blood calcium levels have to be maintained in a homeostatic range because both nerve and muscle function depend on calcium ions. And we're talking about the nerves that allow you to control your activities consciously, as well as your subconscious autonomic functions, your involuntary visceral functions. You also need proper muscle function just to breathe. So if calcium levels are out of normal range, it can lead to death. Calcium is also required for blood clotting, and excessive blood loss and hypovolemia can also lead to death. And many enzymes require calcium as a cofactor. So hopefully you get the idea that normal calcium homeostasis is required for life. The main hormone regulating blood calcium levels is parathyroid hormone, or PTH. PTH is secreted from the parathyroid glands, which are located on the posterior aspect of the thyroid gland, which is located in the cervical region of the body, anterior to the trachea. Parathyroid hormone is released in response to low blood calcium levels. The effect of parathyroid hormone is going to be to raise blood calcium levels in a classic negative feedback pathway, and it does so in three ways. The main target of parathyroid hormone is bone tissue, where parathyroid hormone increases the number and activity of osteoclasts. In doing so, the osteoclasts will break down or resorb old bone. This will release the stored calcium from the bone into the blood, thereby raising blood calcium levels back into homeostatic range. Rising blood calcium levels feed back to shut down parathyroid hormone release. The second target of parathyroid hormone are the kidneys, where parathyroid hormone promotes the reabsorption of calcium from the urine filtrate back into the blood so that less calcium is lost in the urine and it is retained in the body. This also helps to raise and maintain blood calcium levels. Finally, parathyroid hormone targets the kidneys where it activates vitamin D into calcitriol or calcitriol. This active form of vitamin D then targets the gastrointestinal tract to promote the absorption of calcium from the foods that we ingest. So parathyroid hormone, PTH, promotes resorption of bone, reabsorption of calcium in the kidneys, and absorption of calcium in the gastrointestinal tract. Here we have a beautiful depiction of the negative feedback pathway that regulates blood calcium levels through parathyroid hormone. So in this example, some stimulus disrupts homeostasis by decreasing blood calcium levels. For example, the individual is not ingesting enough calcium in their diet, or the client is not taking enough vitamin D and can't absorb calcium. Whatever the disruption is, blood calcium levels begin to decline. This will be detected by receptors in the parathyroid glands, and in response to the low calcium, these glands will release increased amounts of parathyroid hormone. So the parathyroid hormone gene is turned on in a signaling pathway. The body increases the amount of parathyroid hormone. Parathyroid hormone then targets these effectors: osteoblasts in the bone, where their activity and number will be increased. This leads to increased bone resorption and release of stored calcium into the blood. Parathyroid hormone also targets the kidneys, leading to increased calcium reabsorption and activation of vitamin D into calcitriol. That increases calcium absorption from the gastrointestinal tract. Taken all together, these three effects increase blood levels of calcium. And as blood calcium levels rise back into homeostatic range, this feeds back, that's what's detected by that broken gray line, to shut down the release of parathyroid hormone. Now, when blood calcium levels are too high, for whatever reason, two things happen. We saw that rising blood calcium levels shuts down parathyroid hormone, but in children in particular, the thyroid gland will secrete the hormone calcitonin. So thyroid gland secretes not only thyroid hormone, but it also secretes the hormone calcitonin. And calcitonin also targets bone tissue, but here it inhibits osteoclast activity. The decreased osteoclast activity will result in decreased bone resorption, and now there's a switch in the balance between osteoblast and osteoclast activity. Now that there's decreased osteoclast activity, calcitonin indirectly promotes the uptake of calcium out of the blood and deposition into the bone by osteoblasts. So now because osteoblast activity outpaces osteoclast activity, there's an increase in bone building, which requires the uptake of calcium out of the blood into the bone. This essentially lowers blood calcium levels. In the adult, calcitonin is not so active and important. The levels of blood calcium are regulated primarily by increased or decreased levels of parathyroid hormone. We have mentioned several times that bone remodeling occurs in response to changes in physical stressors or mechanical stress on the bone. So exercise and physical activity or inactivity do affect bone strength and density. Under stress, bone tissue becomes stronger through deposition of mineral salts and production of collagen fibers as regulated by osteoblast activity, while unstressed bone diminishes in thickness and density because of the loss of bone minerals and decreased number of collagen fibers. So with bone tissue, if you don't use it, you lose it. The main mechanical stressors are due to the effects again of skeletal muscles pulling on bones via tendons, as well as the pull of gravity. You may have seen that in a infomercial or movie somewhere where astronauts come back after days and weeks in space and they are actually lighter because of decreased bone density. When they're out in space, the pull of gravity is decreased and that decreases the stressor on their bones. Weight-bearing activities such as walking and lifting weights, resistance training, all of those help to build and retain bone mass. Very important as you get older, especially in females, because as I mentioned, coming up, there's going to be a precipitous drop in estrogen levels, and that will then lead to increased osteoclast activity versus osteoblast, and you begin to lose bone density. So one way to counteract that is to build up extra bone density now while you can. We mentioned that sex hormones play an important role in maintaining bone tissue in the adult. As we age, the level of sex hormones begin to diminish in both males and females, and the drop is more pronounced in females after menopause. So what is menopause? It's when the ovaries stop cycling, stop uh, maturing oocytes, and decrease the production of estrogen. It shouldn't really be called menopause. It should be menostop because it's not coming back. Anyways, in response to the decreased levels of the sex hormones, both the androgens and the estrogens, bone mass decreases. And then osteoclast activity outpaces osteoblast activity. Bone resorption is greater than bone deposition, and this is more pronounced in the females. In addition to the drop in estrogen, females generally have smaller and less dense skeletons, so they are already at a disadvantage in that regard. So there are two principal effects of aging on bone tissue. Number one, loss of bone mass. And this results from the loss of calcium from the bone matrix, which is one of the symptoms characteristic of osteoporosis. So with osteoporosis, the again activity of osteoclasts is greater than that of osteoblasts, and the bone tissue becomes porous and fragile and decalcified. Also, in the older adult, bones become brittle, and this results from a decreased rate of protein synthesis. Remember that collagen proteins give bone its tensile strength and flexibility. The loss of this flexibility leads to brittleness and increased susceptibility to fractures in bones that are already weak because of the decreased calcium. Taken together, these conditions contribute to osteoporosis. As you can see, healthy bone tissue in slide A compared to the porous, holy, for lack of a better word, osteoporotic bone on the right. So what can you do to minimize the development of osteoporosis? Maintain good calcium and vitamin D intake, and most importantly, stress those bones. Physical activity into the older advanced years is the best way to slow or prevent osteoporosis. And that's the end of this chapter. Hope you enjoyed that.